Hi everyone, welcome back. Today we will be looking at a 3-point test cross. So let's just get started. A 3-point test cross was conducted, yielding the following progeny numbers for the three mutant alleles X, Y, and Z. Here are the potential gametes for the trihybrid individual in our test cross, and here are the number of progenies that have inherited that gamete. Alright, so in part A, it asks us, what is the genotype of the parental generation? Now, the parental genotypes are usually quite easy to identify, since they are represented by the gamete combinations with the largest number of progeny. In this case, we can see that there are a lot more progeny that have this gametic combination and this gametic combination compared to the other gametes, and thus must represent the parental genotypes. As a result, the parental genotypes will be XX, Y plus, ZZ, and X plus, YY, Z plus, Z plus. Remember that the parental generation is purebred and thus will be homozygous for these traits. Now in part B, we are asked what are the genotypes of the individuals involved in the 3-point test cross. Remember that a test cross is a cross between a heterozygous individual and a homozygous recessive individual. And so, in this case, one of the individuals in this cross will have 2 recessive alleles for all 3 traits, and the other individual will be heterozygous for all 3 traits, our trihybrid individual, and this will represent our F1 generation. And finally, in part C, we are asked to construct a genetic map showing the relative distances between genes X, Y, and Z. So how we go about solving this question is by first determining the recombination frequency between these genes in a pairwise manner, so 2 at a time, which we can then translate into map units later on, as 1% recombination frequency is equal to 1 map unit. Now, it doesn't matter which 2 genes you start with, so we can start by comparing genes X and Y. Now, earlier we determined that X, Y plus, and X plus Y were the parental genotypes for these genes, and so that means that the recombinant genotypes for these genes will be X plus, Y plus, and X and Y. Now, looking at the table, we can see that there are 4 different combinations that have these recombinants, and we will use these to calculate the recombination frequency. Remember that the recombination frequency is the number of recombinants divided by the total number of progeny, and here the total number of progeny is 1,800. With this in mind, we simply add all the progeny numbers for the recombinant genotypes and divide by 1,800 to get 0.487. Now we can then multiply this by 100 to get a recombination frequency of 48.7%. And remember that we can directly translate this into map units, so genes X and Y are 48.7 map units apart. Now we can do the same thing for the next pair of genes. So let's compare genes Z and Y. So again, the parental genotypes were Y plus Z and YZ plus. So the recombinant genotypes will be Y plus, Z plus, and Y and Z. Now we can see that it is these 4 combinations that contain these recombinants, and we simply add these up, divide by 1,800, and multiply by 100 to get 26.8% or 26.8 map units. And finally, the last pair of genes that we will compare is X and Z. So the parental genotypes are X and Z, and X plus, Z plus. So the recombinant genotypes will be X plus, Z, and XZ plus. So we can see that these 4 combinations have the recombinants, and doing as we did before, we add them up, divide by 1,800, and multiply by 100 to get 25.8% or 25.8 map units between these 2 genes. Alright, so based on this information, we can now construct a genetic map. Seeing as genes X and Y are the furthest apart as they are separated by 48.7 map units, we will then put them on the ends of this map. This means that gene Z is in the middle, and it is slightly closer to gene X at 25.8 map units compared to gene Y at 26.8 map units. Now, you may notice some discrepancy here, where 25.8 plus 26.8 is equal to 52.6 map units and not the 48.7 map units which we calculated earlier. But this is simply due to the fact that a double crossover event has occurred within the set of genes, which we will talk about more in the next video. But there you have it, a genetic map with the relative order and distances between genes X, Y, and Z. Alright, that's all for now. Thanks for watching and we'll see you in the next video.